Hi there. My name is Professor Matthew Schmidt, and welcome to Genetics. In this session, we're going to conclude our discussion of the eukaryotic chromosome by talking about the organization and types of sequence elements present in particular, eukaryotic chromosomes. Now, we could have called this section genomics, and the reason I didn't is because it's really not a full exploration of genomics. But we should mention what the idea of genomics is and why this relates to it, and furthermore, why I put this part along into this section about the chromosomes. So, eukaryotic genomes, this field of genomics came about because it's been known for quite some time that the entirety of the DNA in a particular organism doesn't necessarily all have to do with coding for proteins, which is certainly what the gist of genetics was for a long time, and still very, very important. But with the completion of the Human Genome Project, it became very clear that there are all sorts of different DNA elements aside from genes in the genome. So genomics is the detailed study of the entirety of the whole DNA that's around. So a couple aspects of that we can take a sort of quick look at. We're certainly not going to go, you could probably take a whole course in genomics. But one thing is, it turns out when chromosomes are stained with appropriate dyes, and you may have seen pictures of this, various banding patterns are observed. And if you're an expert, you can sort of tell what those banding patterns mean. But one general interpretation that we can say, a lot of times when you see these pictures, they'll be of metaphase chromosomes. But even during interphase, some areas are more darkly stained than others. So the dark staining gives rise to the idea that there are two basic categories or types of chromatin, and they are euchromatin and heterochromatin. Euchromatin tends to be not so densely stained. The density has to do with how much DNA is in that particular region. So the interpretation of the fact that it's not densely stained is that these are single copy genes, and that tends to mean that these are where the protein coding genes are. They're not very compacted, which is how this goes along with our previous discussion, because by and large these genes have to be sort of open because they may be interacting with the machinery of the cell. So euchromatin is not densely stained. If you had to make a real simple one-to-one correlation, we would say that protein coding genes are found in euchromatin or vice versa. Whereas heterochromatin, on the other hand, is very densely stained, which implies that it's compacted, and that also implies that it's an inactive region. Now, that doesn't necessarily mean it does nothing functionally. It's inactive in the sense of it's not getting transcribed, replication's not occurring at that particular point. So usually, and we're going to spend a good amount of time on this, they contain repetitive sequences and not so many genes. So to make a connection of something we've already discussed, centromeres and telomeres are both found in heterochromatic regions. We know that centromeres and telomeres have functions, but generally speaking, they're certainly not something that's getting actively transcribed, for example, all the time. They have a different type of a function. So euchromatin versus heterochromatin. Now, this is about eukaryotic chromosomes, but it's instructive to think about how bacteria are in this respect and then compare that to eukaryotes. So in prokaryote, the genome is very streamlined. It is made mostly of genes. There aren't a lot of extraneous sequences there, and sequences tend to occur only once in the genome. So for prokaryotes, the whole genome, even though the study of genomics is relevant, largely there are only single copy sequences. If you went into this in great detail, we might have to modify that, but that's the basic idea. There's not a lot of what you would call heterochromatin in prokaryotes. You don't even really call what's there euchromatin because it's the only thing that's there. In eukaryotes, we have a whole different situation going on. So there are definitely single copy sequences, but as much as half of the entire genome is composed of repetitive sequences, which can be present in a seemingly ridiculous large amount of copies, as many as 1 million copies. I'm going to try not to say this over and over again, but there's great variability depending on the eukaryotic organism in terms of exactly how much of the genome is made up of this, how many copies are present, et cetera. So we're going to use humans sort of as our organism of choice because we are humans. If you're not, send me an email, okay? Anyway, the field of genomics does study the genome at large in great detail, so we're going to study just a couple of aspects of this that relate to our eukaryotic chromosome idea. So remember, we've got euchromatin and heterochromatin. I'm not saying they go together exactly, but there is an idea that euchromatin has single copy sequences, while heterochromatin probably has repetitive sequences in it. Now, we want to get a little bit of experimental information in here. So how did they even discover repetitive sequences? And we can't go into great detail about this because it would take quite a long time. But there's a process, I think you'll get the idea, that involves denaturation and renaturation of DNA. And the kinetics of that, which is just a fancy way of saying the time frame of how long it takes for denaturation and, in particular, renaturation to take place. So these type of studies really were the way that not only repetitive sequences were discovered, but that different types of repetitive sequences were initially characterized. And also, as you see here, demonstrating their relative abundance in different organisms. So it's a pretty, I wouldn't say simple idea, but you guys are becoming genetic experts now. So in the process, say you take human DNA, you could do it with any organism. You cut the DNA into pieces using restriction enzymes. So you have basically thousands and thousands of pieces of the genome hanging around. You heat it up so that the strands denature. So you have single-stranded pieces of DNA around. And if you cool it down, they start to reanneal is the word, or renature, meaning they find their complementary partner sequences and go back into the more stable double helical conformation. So reannealing, renaturation, the basic idea is we're going to heat this up and let it come back together. But the reason we cut it into a bunch of little pieces is if you just let the whole genome do this, it would not be as instructive for reasons you're about to see. So I hope this makes sense. In general, single copy sequences will renature at a predictable, relatively slow rate. When I say relatively slow, it's because what is fast and what is slow? It's relatively slow compared to some other elements in a eukaryotic genome. But the reason is because, say there's one gene here, if you denature that, first of all, since eukaryotes are in fact diploid, there are in fact 2 copies of each gene, right? But they may not be identical. There are different alleles. But put that little variable out of the picture for a minute. We're thinking that here's a gene, and when it's going to find its partner, its complementary partner, out of all these, who knows how many, millions perhaps of pieces of DNA that are floating around, only one of them is its partner. So the kinetics of this is they sort of all just bump around randomly until they find their partner, and then they renature. So since there's only one correct partner sequence for single copy sequences, it's a slower process as they're trying to look for their partner. So it was found out that certain sequences renature much more quickly than you would predict based on the single copy kinetics that you could, I'll show you a picture in a minute. And it was realized that these sequences represent repetitive DNA. The higher the copy number, the faster it will renature. Because think about a repetitive sequence as opposed to a single copy sequence. When the single copy is looking for its partner, maybe 1 out of 1 million will be able to pair with it. But if you're highly repetitive, I'm just making up the numbers, but this sequence, there might be 1,000 or 10,000 identical sequences around. So you're going to be finding your partner much, much more quickly, right? So it makes sense, and if it doesn't, think for a minute or go back and listen again. Because the more and more identical sequences there are, the more quickly a particular sequence is going to find its partner, and the more quickly that whole type of DNA is going to reanneal in these type of studies. So I know this is a bit scary, but I'm going to attempt to draw a picture. I'm not very good at that. And the graph, the empty graph is here. So time is progressing in this direction. Over here, it's sideways, but I hope you can see this says percent reassociated. Okay, so going from 0 to 100. So here's what I mean by that. If you cut it up in restriction enzymes and denature all the DNA, it's going to be starting up here, right? 0% will be reannealed because it's all apart. And we want to see how fast it goes back together over time. Now, if you were to do this with a prokaryote, a bacterium, and this is not, I'm not good enough to do a detailed graph, but you'll get the idea. So if you took E. coli total genomic DNA, chopped it up, heated it up, and then said, all right, we're going to watch as it reanneals, and don't worry about the technicalities for me anyway. You know, I doubt that you'll have to know this if you do, but there are ways to tell when the DNA has reannealed and when it's still free. We can't discuss that right now. But basically, you'd find that over time, as you cooled things down, you're going to get a relatively straight line as single copy sequences get back together, and it's going to happen over time. The actual amount of time is not particularly relevant because the whole genome in a prokaryote is basically going along in that way, slowly finding their partners. Just by luck, some are going to find their partner faster than others, right? So you just sort of have this line with a reasonably constant slope. Now, thinking about eukaryotes, which I'll draw in blue, maybe when they first did this, they said, oh, we'll have a similar pattern. Now, just for the record, there is a lot more DNA in most eukaryotic genomes than bacteria. I want to superimpose the graph, but the actual amount of time would not be able to be superimposed in this way. But here's the deal. If you do the same type of procedure to eukaryotic DNA, here's what you see. You see three very definite sort of segments or parts of the genome reannealing at different times. So let's make sure we understand what this means. You're starting from the beginning, and all of a sudden, very quickly, a reasonable amount of the DNA gets back together. That's that line going down there. It's like, whew. The shape of my graph isn't perfect, but it gives you the idea. So what these would be over here, these fast reannealing sequences, are highly repetitive DNA or sequences. Okay? Then there's a very definite sort of a lag, and then there's another bunch that go together, and these are moderately repetitive sequences. And then, as you might guess, the sequences that take the longest to reanneal over here are the single copy. So I may have made it a little bit more angular, but it's to make a point that there are really definitely three types of sequences based on their ability to reanneal quickly, pretty quick, or not so quick, all right, in eukaryotes. And you almost always see this. I know I said it before, it's variable. It is in terms of the exact shape of the graph and exactly how much is which type. But that gives you the idea, okay? Now, if you really superimpose this, the prokaryote annealing might be done, you know, by the time we got up to this time frame with the eukaryotes, just because it's taking a lot longer for that many, many more pieces of DNA going together. But what's important is the pattern here, all right? So I guess we should say in red, for the bacteria, it's pretty much all single copy, right? For eukaryotes, only the single copy is down this area, all right? So I hope that makes sense. Let's take a little bit more detailed look at these sequences. So based on these type of renaturation studies, you guys, as you saw, it was determined that there's three major classes of sequences. It's based on their reannealing time, but that's based on their repeat number. Now, just for the record, before we go into that, there are a couple other techniques and terms that you should be aware of. It's hard to know, obviously, for me, because I don't know them, how deep your particular professor or course is going to go into this. So I'm trying to strike a middle ground here. But it's not bad to know these ideas. Another technique that was important in identifying a particular type of repetitive sequence, it turns out that many high copy repetitive sequences collect at a different location than bulk DNA when you place them in a centrifugation density gradient. You may know what that is. Really, really quick, you make, in a test tube, a gradient of something like cesium chloride so that, and you put it in a centrifuge so that things of different densities will go down and they'll find their sort of spot of their density. So you can separate things based on density. So the idea of this is, if most of the DNA, when you do this, is around one area, and then you see another band either at a more dense level or sometimes even a less dense level, this was termed a satellite sequence. Because of their position in the gradient, it's almost like here's the main thing, and this piece up here is a satellite region. That's what they called it. All right, so if you hear about a satellite sequence, you'll know that it is repetitive. It's got a different, it's so repetitive probably that it's got a different density than the rest of the DNA. And one other really important and very cool, I might add, technique is called in situ hybridization. So this allows the mapping of repetitive sequences, but not mapping in the sense that we did with the fruit flies to see where genes are. This is a physical, to actually take physical chromosomes, put probes on there, and see where they bind to the literal physical chromosome. So it allows us to map the locations, and it turns out that they tend to cluster around the centromeres and near the telomeres. But remember, centromeres and telomeres themselves, even though, I mean, we said it, but not in this context, they are repetitive sequences themselves. Like, remember, the telomeres are 6, at least in humans, 6 base sequence over and over and over and over and over again. So this really cool picture shows an in situ probe for telomere sequences, which is easy to make. You know the sequence, you make the complement, and that's the probe. And you see in this picture, I hope you can see, where it's lighting up, that's literally where these sequences exist on the physical chromosome. And predictably, they are at the very tips of the chromosomes, because we said that's where the telomeres are. This is one of the ways that that was established. All right? So very cool stuff, beautiful picture. Let's go on ahead and talk about the types of repetitive sequences. So again, based on the renaturation and sequencing studies as well, it's known that there are definitely repeated sequences in eukaryotic genomes. I know you know there's variability, but in humans, it looks like at least half the genome consists of repetitive DNA, 50% or more. Now, as we just said as well, repetitive sequences fall into 3 categories, but let's be a little bit more specific about what that means. Single copy sequences tend to be 1 copy per genome. Sometimes there are a few, but the idea is very, very small amount, right? Moderately repetitive sequences, it's a little arbitrary, but they say they're present in 10 to 10,000 copies per genome. And the highly repetitive ones, more than 10,000 copies. Sometimes it can be in the millions. So let's take a look at the characteristics of the 3 classes. I put single copies and highly repetitive on the same slide because really there's more interesting to say about the moderately repeated sequences. But single copy sequences, let's really think for a second, because this is not as simple as it might seem. Most protein coding genes, let's just take it there for a minute. Protein coding genes are present in 1 copy, 2 if you take diploidy into account. But if you think of the haploid genome, which is usually the way they do it, there's 1 copy of that gene there, and that's it, right? You got, I mean, if there's a mutation in that 1 copy, it's mutated, that's it. There's nothing to really, another gene there to take its place. There are also regulatory sequences associated with specific genes, and these things exist in the single copy part of the genome. Now, since repetitive sequences are about half your genome, these single copy sequences also comprise about 50% of total DNA in humans. But be really careful, and yes, it's variable. How can you reconcile these two ideas? The Human Genome Project showed that only about 2% of the genome, the DNA sequences, actually consists of coding sequences. I just got done saying 50% is single copy, and then only 2% is actually coding. What is up with that? Well, there's a couple of things you could think about it, but I'll give you a couple of examples. First of all, think about this. In the case of a gene, right? A gene is a piece of DNA usually defined as, it's making one messenger RNA, which is probably making one protein. But think of in that gene, is most of it coding sequences? Well, number one, in eukaryotes, there are usually a lot of intron sequences, intervening sequences. They're part of the gene, they're even part of the initial messenger RNA, but they're not coding sequences, right? I wouldn't say they're rubbish, but in this context, they're not coding. They have to be removed from the gene or from the transcript before it can be translated, right? So even though that's part of a gene and it's present in a single copy, it's definitely not coding. And you may remember as well when we talked about RNA that a big part of a messenger RNA consists of a 5' untranslated region, a 3' untranslated region. There are poly A tails, that doesn't count exactly, but the point is that a lot of the DNA that's in a gene doesn't actually end up in a finished messenger RNA, which is truly the coding sequences, right? If I say variable one more time, you're probably going to get mad if you're not already. But, it depends even in one organism, from one RNA to the other. There may be more introns, there may be more non-coding parts of the RNA. Even the regulatory sequences such as the promoter, eukaryotic promoters, we didn't talk a lot about it yet. You may have in your class, but they can be quite large and they can be present in single or a few copies. So all of this stuff, as it were, even though it's single copy, it's not necessarily literal coding. Coding in the sense that here's AUG, that means methionine. So be very careful, even if you say genes versus coding, actual coding sequences, okay? So I hope that makes sense. Now the highly repetitive sequences, the best example of these are actual telomere and centromere sequences. They're not the only ones, but since you already know something about those, they're good ones to think about. Telomeres, 6 bases over and over and over and over and over and over and over again. Centromeres are longer, but they're repetitive for sure. So they're in this part of the genome. Also, satellite sequences, as we just implied, are composed of this highly repetitive DNA. Sometimes we have no idea what the function of these are, or even if there is a function. I really hate this term, and I hope that it's going out of style, but some people call repetitive sequences that they don't understand junk DNA. What used to be junk conceptually may very well be turning out to be important. We see all the time things that were not understood before in terms of their function do perhaps have a function. But the moderately repetitive sequences are perhaps the most interesting because there are all sorts of different sequences in here. So starting from, we said that they were 10 to 10,000. Some genes, I know we said this was not the case, but this is the exception to the rule. A perfect example of genes that are moderately repetitive are ribosomal or rRNA genes. The reason is you need a lot of ribosomal RNA. Cells have sometimes thousands of ribosomes in them, all constructed of ribosomal RNA. And they tend to be present in tandem copies. So tandem just means one after another. Say there's like 10 ribosomal RNA genes present. The real reason for that is, in theory, you can amplify lots of mRNA and proteins off of one copy of a gene, but if you really want to get a lot done fast, the more copies of the gene you have, the more transcription and translation can occur. So rRNA genes are present in tandem repeats, not a lot, but usually about 10, let's just say. Maybe more than 10, but not like 1,000. So those are actual genes. Now they're not protein coding genes, right? They're RNA encoding genes. All genes encode RNA, right? It's just a matter of whether they encode messenger RNA, which will then be translated, or if it's just RNA that has a direct function, as in this case. And you may know, or if you don't, let's say it. The nucleolus, so-called, is not really a structure, but it's a place where all the ribosomal RNA genes exist, and they're usually being transcribed. There's a lot of activity going on there and a lot of nucleic acid being produced, so it stains very darkly, and that's what the nucleolus is, or nucleolus. But the rest of this, and most of it, are sequences that are either transposons or sequences, probably more of them, that are derived from transposons. So what the heck is a transposon? You could have multiple lectures on this. But transposons are sequence elements that are able to copy themselves and then reinsert a copy of themselves into another portion of the genome. Some people call them jumping genes. I don't know if I would even call them genes, but they jump. Now, a lot of transposons are active in your cells of eukaryotic creatures. It almost sounds a little creepy, like there are viruses running around, and in fact, probably viruses evolved from transposons somehow or another. But the point here is more that the fact that transposons have an over-evolutionary history, copied themselves over and over and stuck themselves into different places in the genome, would certainly explain why there are a bunch of copies of them hanging around. So there are some active transposons, but there's also a lot of not inactive transposon-derived sequences around in here. We can tell by the sequence that it's from a transposon, but it may not be actively copying itself at this point. So about 45% of the total DNA in humans is from this. So what this means, guys, is that if 50% was repetitive or heterochromatin, right, it means that even though highly repetitive sequences are, well, highly repetitive, that this part, the moderately repetitive, really makes up the bulk of the repeated sequences. So if you really get into this, there's a bunch of, it can get really complicated, but I wanted to just break it up into two sort of major categories, the lines and the signs. These are, lines is a acronym, is that the word? For long interspersed nuclear elements. All it means is that they're interspersed is sort of important, whereas ribosomal RNA genes are tandem, one after the other. These are not necessarily in any easily predictable location. One's here, one's there, one's over there. So they're interspersed, they're relatively long. The length on average is about 6,000 base pairs. And they say that this makes up about 20% of the human genome. Signs, as you might guess, are short interspersed nuclear elements. And the difference is simply that they're shorter, average length of about 300 base pairs. They say about 15% of the human genome is comprised of this. A very famous type of sign that you probably do have to know about is called an ALU repeat. Now, these can be present in over 1 million copies in human genomes. It's striking. It's still not fully understood why they exist. So this is about 10% of the whole genome just of the ALU repeats. Now, a couple things. You might be saying, what the heck are you talking about? If they're in 1 million copies, why aren't they, you just said that these are from 10 to 10,000, right? Why aren't they highly repetitive? The reason is, guys, that the ALU family has a lot of related sequences, but they're not identical. So if you have 300 base pairs, the whole reason they're called ALU is because there's a restriction enzyme called ALU1, I think it's ALU1, that was found to cleave all over the place in these regions, cut. So they called it the ALU repeats because that restriction enzyme cut it before they knew about the sequences. But just because ALU cuts it, that means its recognition sequence is there. It doesn't mean that the whole sequence is identical. Now, don't get me wrong, these probably all arose from either one or several related, genes originally. I said genes, but I mean repeats. So the ALU repeats, yeah, they're very, of all the things in your genome, this is probably the largest, most interesting family of repetitive sequences. So, I mean, there may be 1 million copies of ALU relatives, but each particular element wouldn't be present in more than 10,000 copies. Well, obviously there's a lot more you could learn about this, and if you're interested in genomics, I think that's great. You should learn more about it. But this is what I think we should do to wrap up our section on the eukaryotic chromosome. I hope you liked it as much as I did, and I'll see you real soon.